including the enzymes at each step and the key steps of ATP gain or release. So the glycolytic pathway starts with glucose. So glucose is first converted into glucose 6 phosphate under the action of hexokinase, which is the enzyme here. So it's very important to remember the enzyme in each of the step of any biochemical pathway, as enzyme plays the important role to convert the substrate into the product. The next step converts glucose 6 phosphate into fructose 6 phosphate. So you might be wondering what's the need to convert glucose 6 phosphate to fructose 6 phosphate. So if you look at the structures of glucose 6 phosphate, it's asymmetrical, and so it's not easy and it's not easy to cleave. Rather, fructose 6 phosphate, when phosphorylated, can be cleaved easily into 2-3 carbon molecules, which can be further oxidized to get pyruvate. So this is a better choice because of the symmetry and stability. And this reaction takes place under the action of the enzyme phosphoglucose isomerase. So one of the key advantage in biochemical pathway is that all the enzymes fall in either of one of the in a predefined fixed category. So if you understand the reaction, you should be able to give the name of the enzyme. Watch my enzyme class video to know more about the type of the enzyme and how you can actually just guess the name of the enzyme for a particular reaction. So the fructose 6 phosphate is further converted into fructose 1,6 bisphosphate, and this reaction takes place under the enzyme phosphofructokinase. So just a hint, whenever there is kinase in the enzyme, the substrate is always going to be phosphorylated. Phosphorylated means that a phosphate group is gonna be attacking the substrate. So just a hint, whenever you have the word kinase, substrate is phosphorylated. So fructose 6 phosphate is then converted and fructose 1,6 biphosphate. So one other noteworthy thing of this reaction is this is the first step where an ATP molecule is consumed and you get ADP. So remember that we are still in the stage 1 of glycolytic pathway, and stage 1 is the energy consuming stage. So here one ATP is consumed, and in the very first stage, when glucose is converted to glucose 6 phosphate, one molecule of ATP is converted into ATP. So these two steps are very important, as these are the steps when ATP is consumed. So if you see till now, you have minus 2 ATP, as you have used 2 ATP molecules and you haven't actually produced or yielded any ATP molecule till now. So after the fructose 1,6 bisphosphate, under the enzyme aldolase, it is you get two intermediates. One is glyceraldehyde 3 phosphate, glyceraldehyde 3 phosphate, which is also known as GAP, and the other one is dihydroxyacetone phosphate, which is known as DHAP. And these two intermediates are interconvertible. It means that they can spontaneously change from one form to another without any need of energy, and it's under the enzyme isomerase. So you must have noticed that this is the second time we are using the enzyme isomerase. So isomerase is an enzyme which is used at the stages when you want to convert when you do for in the interconversion of two isomeric molecules. Well, isomers are basically structures with the same chemical formula but different structures. I'll link down the video to about isomerism and isomerase, basically, and to if you want to know more about it. So GAP is the only intermediate which can be used in the further steps of ATP. So GAP is then converted into 1,3-bisphosphoglycerate under the action of the enzyme glyceryl 3-phosphodehydrogenase. So one key easy way to remember the name of the enzymes in any biochemical pathway is look at the substrate, and most of the time the name of the substrate would be in the prefix part of the enzyme name, whereas the later part of the name will focus on what kind or what class of enzyme it would be in. So this one is dehydrogenase. For example, another class is isomerase. Then kinase is also one of the classes of enzyme. So just look at I'll link down my enzyme classes video, and you can look there and you can watch the video for more details. So after the 1,3-bisphosphoglycerate conversion is also an important key step, as in this step NAD plus is converted to NADH. NAD plus is also one kind of energy carrier, and we'll learn about more this, and we'll learn more about this in the following videos when we go to oxidative phosphorylation. So for now, when you have 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate, and this is the step where this is the first energy yielding step till now, where ADP is converted to ATP under the action of the enzyme phosphoglycerate. So this is the prefix, the front part of the name of the enzyme, so phosphoglycerate kinase. And if you are able to correlate and you know formal logic by now, you should know whenever kinase is there is a phosphorylation happening. So in this one, you have two phospho group attached to the glycerate and in the substrate, and in the product you only have one phospho. So one of the phosphorus group is transferred to the ADP. So what actually is ADP? ADP is nothing but adenine group with two phosphate group, and ATP is 3. So one of the phospho group from 1,3-bis phosphoglycerate is transferred to ATP, and in the end still and one of the phosphate group remains attached in the product. From 3-phosphoglycerate, further along the line, under the action of the enzyme phosphoglycerate, so note the similarity and the pattern which the enzyme name is following. It will help you remember the name of the enzyme. So phosphoglycerate mutase converts 3-phosphoglycerate to 2-phosphoglycerate. So if you can see, basically the position of the phosphate group is changed. Here, the and ending in this one, the phosphate group was attached at the 3 carbon position of the glycerate, whereas in this one it is attached at the 2 carbon position. From 2-phosphoglycerate, under the action of the enzyme enolase, we get phosphoenol pyruvate, which is also known as oxaloacetate. So we are almost towards the end of the glycolytic pathway. So from oxaloacetate, under the action of pyruvate kinase, so again see the pattern kinase. So it should actually so it should actually pop up in your mind that kinase is there, so there must be a phosphorylation happening somewhere. And phosphorylation is basically transfer of phosphate group from a substrate to the intermediate, or substrate to the enzyme, or substrate to the product. So in this one, this is the last step, and it's an energy yielding step, because an ADP is converted to ATP, and hence the kinase class of enzyme is justified. And so from phosphoenol pyruvate you get pyruvate. So pyruvate is the end product of glycolysis, and depending upon the environmental depending under the environmental condition and the bridge reaction, pyruvate will either be converted into lactic acid or completely oxidized to ATP, or there would be alcoholic fermentation. So the overall reaction of glycolysis can be summarized as glucose plus 2 ADP. These 2 ADP molecules were used in the second stage of glycolytic of the pathway, plus 2 pyrophosphate and 2 NAD plus, which gives 2 pyruvate plus 2 ATP plus 2 NADH. So this is the overall glycolytic pathway. But the key thing here is that after this point, when you have GAP is interconvertible under the isomerase to DHAP, this all, like up to the production, all this runs twice. This cycle runs twice. This is because from fructose 1,6 bisphosphate you get GAP and DHAP. But in this under cellular condition, DHAP is actively converted into glyceraldehyde 3-phosphate. So you have 2 glyceraldehyde